Hi everybody, this is Mrs. Mancuso and I'm going to go through the Cells Big Idea sheet to help you prepare for the AP Bio exam. So here we have an animal cell over here and a plant cell over here and a whole bunch of labels. And so down beneath we've got to write about each one. So I know I'm not going to be able to write in this little tiny space. So I'm going to talk about each one and you guys are going to be filling it in. Again, you can be checking my answer key to make sure that you got it all. So A right there, that's the cytoplasm. So the cytoplasm contains a lot of enzymes. It's definitely a supportive environment for the inside of the cell holding each organelle. But there are some reactions like glycolysis that occur in the cytoplasm, so it definitely does contain enzymes. All right, B. B is the chloroplast, and you see it just in the plant cell. This area right here that I just did the little red dots in, that's the grana, and the grana are the stacks of flattened membranes. They have thylakoid membranes, and they have lumen as their interior space. That's where the light reaction happens. And then here, in that space is the stroma, the liquid part of the chloroplast. So B is the chloroplast, and that is the site of photosynthesis. C is pointing to the nucleus, and the nuclear membrane is what surrounds the nucleus. It's a double-layered membrane that has pores that allow RNA out but not DNA. So they're small so that DNA is protected. And then D is the actual DNA. So the DNA are it depends on the type of cell, but they're usually arranged in chromosomes, which hold the genetic information, which means they have the directions for building proteins. All right, E is ER, and it appears very smooth right here. So smooth ER makes lipids. Over here, where there's a little bit of ribosomes on it, that is rough ER, and rough ER transports the proteins that are made by ribosomes. F, first of all, let's cross this right out because that is super large and it doesn't look like these ones. So these little dots are ribosomes, and ribosomes are the site of translation. So this is where proteins are made. All right, G is a Golgi body, and a Golgi body is made to modify proteins, which means it alters their structure a little bit and prepares them for whatever they're going to be. They usually go from being made at a ribosome, transported by ER, into a vesicle, into a Golgi body where it's modified, into another vesicle, and then out. All right, H is pointing to a vacuole. And I would say we could have another vacuole over here inside the animal, just a much smaller one. So H is our vacuole. It's for storage of different molecules, especially in a plant. If they're making excess sugars, they would get stored there. If it was a CAM plant, they could store malic acid, which is their form of carbon dioxide, at night. But in most plant cells, it's just really large to exert a turgor pressure to keep the shape of the overall plant cell. All right, I would say in the animal cell, I is pointing to a lysosome because it clearly shows that there's something within it. So that contains digestive enzymes to help break apart whatever's entered a cell. And in plants, it's not really likely to have a lysosome because they don't digest. They produce their own sugars. So over here, it's just odd, the placement of it. So let's cross it right out of the plant cell. All right, J. J is pointing to the cell membrane. And the cell membrane is a phospholipid bilayer that is selectively permeable to allow things to enter and exit the cell. K is mitochondria. You always notice because it has this double membrane. The matrix is this very middle part. That's where the Krebs cycle happens. And then out here's the outer compartment, and the squiggly membrane is called the cristae. So this is the site of aerobic respiration. It's where ATP is produced, and the Krebs cycle and oxidative phosphorylation both happen here. All right, now let's go down to the next question. So what are the key differences between prokaryotic and eukaryotic cells? So if you have a prokaryotic cell, the main difference is it has no membrane-bound organelles. So a membrane-bound organelle is something that has a phospholipid bilayer around it, like a mitochondria or a chloroplast or a nucleus or a vacuole. So a prokaryotic cell does not have that. So if it doesn't have that, what kinds of things could it have? It can have ribosomes. It can have cytoplasm. It can definitely still have its DNA. It has its outer cell membrane, just doesn't have any interior ones. All right, and then over here, a eukaryotic cell has membrane-bound organelles. So it's more advanced, and it formed by endosymbiosis, which means an original cell took in another cell. So we've got a prokaryotic cell and a prokaryotic cell. And once it engulfs it, it's going to look like this. And that, actually I need even a layer of red around it. It's going to look like that because it surrounded it, engulfed it. So an example, this could be a mitochondria. A mitochondria used to be its own cell, and some proof of that, it has a double membrane. One of the membranes matches the cell membrane of the cell it's in, and one is different, and that's different because it was originally a different cell. It also has its own DNA, so we know that chloroplasts and mitochondria both used to be their own cells. All right, why is compartmentalization important? That's because it separates the enzymes that are required for different reactions. It also is going to separate your reactants and your products for different reactions so that they're not all in the same place, so that they're more likely to be protected and undisturbed. Nucleus. So I already talked quite a bit about this, that it has a double membrane and that it contains DNA and it has pores. So I'm going to move right on to the next one. What are the components of the endomembrane system? Imagine a protein is to be exported. So a protein is produced at the ribosome, and it moves from the ribosome into rough ER, and the rough ER transports it, usually into a vesicle, and that vesicle to a Golgi body, and at the Golgi body it would get modified and changed, and then into another vesicle, and then eventually out the cell membrane by exocytosis. All right, describe the following types of junctions. All right, so I'm going to go ahead and create a new page. All right, a tight junction looks like this. Here's the edge of one cell. Here's the edge of another cell, so I can complete it and I can say, all right, these are two cells. Tight junctions look like grout between two cells. They really closely join the two cells so that any liquid that's over here on this side can't get through. It can't pass. So this is going to be important in places like your stomach. All right, the next one on that list is gap junctions. Okay, so this is what a gap looks like. All right, it's basically a channel between two cells so that molecules can freely move between the two cells and they can communicate quickly. So it's like a channel joining adjacent cells. And then the next one is desmosomes. So here's one cell, here's the adjacent cell. They're fibers that extend through so that no matter how much you pull, you can't separate the cells from each other. So if you go ahead right now and pinch some of the skin on your hand and you can lift it up in the air, your cells don't separate from each other and come flying off your hand into your finger. It's because they're all connected with desmosomes. And then the last one on that list is plasmodesmata. And plasmodesmata is a gap junction in a plant cell. Sorry, I lost my place. This happens all the time. Okay, plasmodesmata, gap junction in a plant cell. All right, so I'm going to use that A, B, C, D that I had above. So found in a plant cell would be D, plasmodesmata. Forming a channel between the cells would be B and D, so gap junctions and plasmodesmata. Allow free passage is also B and D. Block intercellular transport, that would be A, a tight junction. Found in layers that separate two solutions, that would also be A. Lining of the digestive tract, that's also A. Riveting cells together is C, my desmosomes. Well developed in cells subjected to mechanical force, that would be C, desmosomes. Permit intercellular transport would be B and D. Glycoprotein filaments penetrate, that's going to be C, my desmosomes. Animal cells have A, B, and C. And hold cells tightly together would be C, desmosomes. All right. Next, we're moving on to cell membranes and transport. So here we have a nice picture. I know mine is in color and yours is black and white, but let's talk about all the things that we have. So first of all, this is just one phospholipid. And remember, phospholipids are amphipathic. That head right there is polar, and the tails right there are nonpolar, which makes the tails space into each other, and it makes a bilayer. And then this molecule right here, this is cholesterol. That's going to increase the flexibility and the movement because it gets in the way of the tail. So it makes it a little bit more flexible of a membrane as long as it's not too cold out. All right, and then any of these large purple molecules, those are proteins, and proteins act as channels or receptors or recognition molecules within a bilayer. So they are also amphipathic if they go the whole way through, because this area of it must be nonpolar to be next to those tails, and this part must be polar to be next to those heads. So you could also say that this channel right here is amphipathic. So this one it tells me is a gated channel. So if it's a gated channel, ions can move one direction or the other. It usually is just a one-way thing. But this gate would have to be open for it. So usually there's some sort of signal ligand that causes it to open and ions can move in and out. Ions can't get through a bilayer because ions require a protein. They have a charge. So a plus or a minus will not be able to get through this. You got to be small and nonpolar to get through the lipid bilayer. All right, here is a channel protein. It doesn't have a gate, so it's always open. So it moves things from high to low. All right, a receptor has that specific shape right there that we see. So that is for receiving something. It could be a neurotransmitter. It could be a ligand, some part of a signaling pathway. All right, this one, the transport protein, this one looks like a pump. So this one actually has to change conformation, change shape. That means it's going to have to bind ATP. It's a type of active transport. It's going to have to open this part up. So it's going to have to change shape so that this opens like this and allows the molecule through. All right, up here, this carbohydrate, this is making up your MHC. So this is a recognition protein. So recognition proteins allow you to be able to tell self versus non-self cells. All right, so let's go through this together. If I have a receptor protein, it doesn't allow anything to enter. It just receives a ligand. And examples of molecules, well, they're not going to enter or exit, but molecules that fit in it, so I'm going to put ligand fits, and this is for a signal transduction pathway. All right, a channel protein is definitely facilitated diffusion. And examples of molecules that go through a channel are ions, and they go from high to low. A gated channel protein is also facilitated diffusion. It also would be ions, and it's also high to low, but it has a gate, so it requires a ligand. All right, a transport protein is usually active. These are what we call our pumps. and they also can move ions, but they're going to move it from low to high. So if you want to stockpile something against the gradient, that's going to require a transport protein or a pump, like the sodium-potassium pump. All right, a glycoprotein is for recognition. It is not for diffusion or active transport. And so nothing enters or exits because of it. And special features, it is able to tell if your cell is self or non-self so that you know if you should attack it or not. All right, MHC is our overall glycocalyx, our sugar coating covering that is unique in its markers to identify self versus non-self. All right, we've got next 2. Water moves from an area of blank water potential to an area of blank water potential. Well, that's easy. High to low. Things always move high to low. All right, define the terms hypotonic, hypertonic, and isotonic, and would explain what would happen to a plant cell and animal cell in these solutions. All right, so if this is my cell and this is my solution, if the solution is hypotonic, that means the solution has more water and less solutes. So that means I need to have a higher concentration of ions inside the cell than outside of the cell. And what happens is the hypo is going to lose water. So I'm going to draw an arrow showing water going this way. So I've got H2O going into the cell, and the cell is hypertonic, and the goal is to get to a state of being isotonic or balanced in concentration on the inside and the outside of the cell. If you put a plant in a hypotonic environment, it is very happy. So for a plant, ideal is hypo for the solution. That way the plant cell's always gaining water. All right, here is another solution, and I'm going to call this solution hyper. So if this is my cell and it's in a hypertonic solution, the hypertonic solution has more ions, it's more highly concentrated than within the cell. So my cell has to be hypo. So these words only work when comparing one solution to another. Water always moves from the O to the I always say, think of the O as a kid with a big fat water balloon, and he's ready to throw it at a hyper kid. So the hyper has more sugar, so water goes from hypo to hyper. All right, next one. For the following diagram, 2 solutions are separated by a semi-permeable membrane that's permeable to monosaccharides but impermeable to other carbs. So side A has 7 fructose and 3 glycogen. Well, right away I'm going to put a cross on the G because I'm not moving glycogen. It's not permeable to glycogen. And then over here on this side, I've got 4 fructose and I've got 2 glycogen. I'm going to put a cross over it showing that I can't move that. So at the initial point, this side adds up to 10, this side adds up to 6, which means this side has more solutes, so this side is hyper and this side is hypo. So which side is hypertonic at the start? A. All right, which side has a higher water potential? That means there's more water and water will move from it, so B has a higher water potential at the start. Which side will water move to? So again, I don't want to move the water first, I want to move the sugars first as long as it's permeable. So I've got a 7 here and a 4 there. 7 and 4 add up to 11. I'm dividing it by 2 sides. That means it's going to even out at 5.5 each. So this is going to go up to 5.5, so I've got sugar moving this way. So I've got fructose moving to this side. 7 is going to go down to 5.5. And now that I have them at 5.5, I can re-add. So now I'm at 8.5 on this side, and I am at 5, 6, 7.5 on this side. So it's still hyper over here. It's still hypo there. So that means water is going to move from B to A. So as water moves over, you are going to see the level over here go up, and you're going to see the level over here go down. So water is going to move to side A. What happens to the glycogen concentration? They usually even out. They usually become pretty isotonic. The glycogen isn't moving, but you're changing the water around it. All right, next one. What is facilitated diffusion, and give examples? So we already talked about that quite a bit. That means that you are allowing a channel to move something in or out of a cell. It's usually something like an ion, and it is passive, so you are moving it from a high to a low concentration. And why do some cells need active transport? Usually because they're going against the gradient. So if you want to go from low to high, and that definitely happens with calcium in a muscle cell. It also happens with sodium and with potassium, with the pump at the end when you're trying to get back to a resting state. All right, how can a concentration gradient across a membrane be a source of potential energy? All right, so what you can do, if this is my cell, within my cell I could have chloroplast. So in here is a chloroplast. Oh my gosh, I have to draw so small. So there's my grana. Within my grana, I could have photosystems that pass electrons through an electron transport chain. And in that electron transport chain, as the electrons move, they move H pluses. So electron transport chain, as it passes electrons, the energy is used, because those electrons are high energy molecules, the energy is moved to pump H plus against its gradient. So you end up building up a whole bunch of H pluses in one spot. All right, so these are all H pluses. That's my proton gradient. Those are my hydrogen ion gradients. And then you have to use this special molecule. It's got a rotor on it. It's called ATP synthase. And as you move these from high to low, this will spin, and it will connect ADP and PI, and it will produce ATP. That process is called chemiosmosis. All right? So this is all for number 7. So now that we have number 7, sorry, but I'm going to go ahead and erase this so I have a little bit of room to read number 8. All right, number 8 says, explain how the sodium-potassium pump generates a membrane potential or an electrochemical gradient. So a sodium-potassium pump, what it does is, here's my axon in a nerve, and I already had sodium come rushing in, and I had potassium go out, so this caused depolarization, this one caused repolarization. But the problem is that now K plus is out here and Na plus is in here, and if I want to get back to rest and so I can send another message, I need a pump. So this pump is going to move Na plus back out, and it is going to move potassium back in. So I'm going to put another K plus here, and it's going to move it in. So that's how it works. And it can restore the negative 70 millivolts that exist. It can repolarize a membrane and get it back to a charged state. Alright, so that is the end of this video. Make sure you take some time to go over it and write down any questions that you have for me.